This lecture will be an introduction to microbiology for the surgical technologists. The learning objectives for this chapter will be to discuss the responsibilities of the surgical tech in prevention of disease transmission, discuss significant historical contributions from pioneers in microbiology, relate notable discoveries and events from the historic timeline to current concerns, compare and contrast the theories of spontaneous generation and biogenesis, and lastly, discuss the scientific impact of Koch's postulates, including the exceptions to them. As a surgical technologist working in the operating room, we want to make sure that we give the patients the best possible care. And one of the ways that we do that is making sure that we understand what is sterile and what is not sterile, because that is what's going to keep our patient from getting a surgical site infection. And as of 2017, surgical site infections in the U.S. totaled between 160,000 and 300,000. And the estimated cost, the healthcare cost, is between $3.5 billion and $10 billion. And so it's really important, not only from a monetary standpoint as far as what we're costing the patients, but their time, how much time they're going to spend in the hospital, and pain and suffering and all of those things, because good sterile technique is the best way that we can protect our patients as surgical techs. So agere primo, the patient first, that's what surgical technique is about. And one of the ways that we can have a better understanding of sterile technique and what's required of us is by having an understanding of microbiology. Okay, so now we're going to cover a little bit of history, so stay with me now. We're going to start by talking about a gentleman named Fracastoro. And in 1530, Fracastoro published some research regarding syphilis. And then in 46, he published these findings in a paper he called On Contagion and Contagious Disease. And his theory was that there are tiny little unseen creatures, and these are spread by means of contact between an infected host and others, indirect contact, carried on clothing, or carried through airborne transmission. Sound familiar? He might have been on the right track. However, following his death, his beliefs kind of fell out of favor and didn't really resurface until the 1800s. Now, Robert Hooke, an interesting English scientist, he designed and built a microscope, and you can see him here in this image looking at a mammoth. And you may have also heard something called Hooke's Law. So Robert Hooke also developed Hooke's Law. Anyways, at first when he developed his telescopes, he was looking at heavenly bodies that, in the sky and those kinds of things. And in 1665, he published a work he called Micrographia, which means small drawing. And he was the first one to use the term cell, but not for a cell like we know it today, but he looked at cork under a microscope, and he saw these very small honeycomb-like spaces. And so that's what he was referring to as a cell. However, this was the beginning of what we now call cell theory, and that all living things are composed of cells. Now, Anton van Leeuwenhoek was an amateur scientist, and he was the first to observe and record bacteria and protozoa in 1673. And so he was using a single lens microscope, and he looked at rainwater and saliva and even his own semen, and he saw these little creatures, but he referred to them as animalcules. and he reported his findings, and he made the first accurate drawings of these types of bacteria, and they served as the basis for the modern depiction of the previously invisible world that couldn't be seen with the naked eye. And then lastly, Francesco Redi, even before Leeuwenhoek reported his findings, he was trying to disprove a theory called abiogenesis. And abiogenesis is the theory of spontaneous generation. And this means that the thought was that life could spontaneously arise from any type of non-living material. But he wanted to disprove that. He was openly critical of it, and so he did this experiment where he took 6 jars, and in 3 of them he put some meat and he left them open, and the other 3 he put some meat and he closed them. And after a few days, maggots appeared in the open jars but not in the sealed jars. And so he did receive some criticism from this. The other experts in his field were saying, well, it didn't disprove abiogenesis because there was an absence of oxygen in the sealed jars, and that's what is required for spontaneous generation. So he said, okay, I'll do the experiment again, and he did. And but the second time, the 3 jars that he had previously sealed, he put a fine mesh over the top of them so that nothing could get into it. And guess what happened? The same thing happened. There were maggots in the open one and nothing that could be visibly seen in the ones that were covered with mesh. So this provided a strong basis for the refuting of abiogenesis by the scientific community. They weren't ready to give up that theory yet, so it would be a little while longer before they would come to the same thinking that Redi had. Continuing on with our historical account of microbiology, comes on the scene John Needham and Lazzaro Spallanzani. And John Needham was this English priest, and he had performed some experiments in the mid-1700s with some meat broths, and they became infected with microbes. And so his take was there was some vital force that was responsible for this spontaneous generation of life. And then Spallanzani came along and said, no, that's not the case. And he did a whole bunch of other experiments, but he was never really successful of kind of swaying the scientific community that, spontaneous generation just wasn't a thing until and then in 1798, Edward Jenner actually started working on the first vaccination, right? And so in the late 1700s, smallpox had become a big deal all over the world, and many people were dying, and they had tried something similar to vaccinating individuals with some fluid from the pustules of the infected host, but it didn't work, and oftentimes it made him really sick. And then he had this really great mentor. His name was Hunter, and he said, why think or speculate? Why not try the experiment? And so Jenner took this to heart, and so he observed that individuals that got a much less serious disease called cowpox, they would easily recover, and then they wouldn't contract smallpox. So he got this idea that he would inoculate an 8-year-old boy with some fluid from the blisters of a milkmaid that had cowpox, and he became mildly ill and he recovered. So then Jenner got this idea that he would inoculate the same boy with smallpox, and the boy never developed smallpox. And years went by, and it wasn't until 1823 that Jenner really got the accolades that he deserved in providing immunity through vaccination. And then we come to our dear friend Semmelweis. Remember Semmelweis and his observations regarding puerperal fever? In the early 1900s, puerperal fever was taking a lot of moms' lives, right? 25 to 30%. And so Semmelweis, if you remember, is the one who observed the midwives and that they would wash their hands between patients, unlike physicians and med students who would go right from the autopsy room into the exam room and examine their patients. The midwives were careful to wash their hands between each patient. And so he got this crazy idea that hand washing actually reduced the spread of disease. And so he became very adamant about hand washing. Other physicians thought he was cuckoo. And because of that, Semmelweis actually had a mental breakdown and interestingly ended up dying in 1865 from a surgical site infection following a small procedure that he had. Louis Pasteur and Joseph Lister were two of the most recognized figures of the golden age of microbiology. Like, the things that they discovered were so amazing, and a lot of those practices, of Lister's practices and Pasteur's practices, we still do today. So Louis Pasteur, he was a French scientist, and he conducted some experiments with some flasks and some beef broth. And long story short, what he ended up determining was that when he had flasks that were just open to the air, they did develop microbes, but then he used ones that had a little crooked neck and they didn't develop microbes. And so he was like, hey, maybe that's because, you know, the little crooked neck doesn't allow particles from the air to get into the beef broth. And so this was the first step in eliminating the abiogenesis theory of spontaneous generation of life from nothing. And so he was able to dispel Needham's vital force theory and really started the whole thinking for the germ theory of disease. All right, that microbes might actually cause illnesses. No way, you don't say. So what else did he do? He picked up on the fact that bacteria were responsible for the spoilage of beer and wine, and that bacteria changed alcohol into acetic acid, which is vinegar. He also discovered that bacteria could be classified as either aerobic and anaerobic, so some have to have air and some don't want any air at all. And then the thing that's we probably hear about the most with Louis Pasteur is pasteurization. So his thought was, you know, to heat a solution just enough to kill the bacteria, and then you wouldn't impair the taste or the product. And so that is the same process that we use today to kill bacteria in milk, is pasteurization. And so again, you know, he was a huge contributor of the establishment of the relationship between diseases and microbes, and that is pretty amazing. And then later on in his career, he did some work with rabies, the rabies virus. And so there came a time when there was a young boy that had been bitten by a rabid animal, and he was approached and asked if he could vaccinate the young boy. Well, he wasn't a doctor, he had never really done anything like that, but he took a risk and he did vaccinate the young man, and he did recover and survive. So that's pretty amazing. And then Joseph Lister, he was an English surgeon, and he is responsible for establishing some of the first principles of aseptic technique. Like, these are the ground rules that we use for surgery. He was also responsible for the first preps and skin scrubs that we used. He understood that carbolic acid killed microbes, and so he would wash his hands with that. He would have the patient prepped with that. He also had it sprayed over the surgical field to prevent patient infections, and he was also a big supporter of wearing gloves, changing gowns and aprons between cases, and cleaning and disinfecting surgical instruments by boiling them before they were used on the next patient. Thank you, Joseph Lister. So now we're going to spend a little bit of time talking about Robert Koch. And Robert Koch was a German physician, and he is named the most influential bacteriologist in the history of medicine and epidemiology. That is pretty significant. So he studied under Henle, and Henle was a German anatomist, and he flourished under Henle. Henle believed that disease was caused by microorganisms, and this influenced Koch to do his research, and he started with research regarding Bacillus anthracis, which is the bacteria that would cause anthrax in cattle. And so he cultured that bacteria and he injected it into healthy animals. And when the animals became sick, he cultured that. He isolated the bacteria and examined the two, and he identified that it contained the same bacteria. So thusly, he developed these practices that we use for conducting experiments regarding specific microbes causing specific diseases. And so these were called his postulates. And so in 1877 was the first time that his 4 postulates were published, and this is what they said. The first one said the same disease causing microorganism must be observed in all cases. All right, so for it to be there to be a correlation, right? And that all pathogens must be isolated and grown in a pure culture. If it's contaminated, it's not going to work, right? We're not going to get the same result. The pathogen, number 3 is the pathogen from the pure culture must reproduce the disease when inoculated in a susceptible animal, right? So if you're going to isolate something, you've found that it did something in one organism, it has to do the same thing in another organism. And the last was the microorganism must be isolated from the inoculated animal and proved to be the original disease-causing pathogen, right? So they had to be the same. And there are some exceptions. One of the exceptions is not all microbes can be grown in a lab. They have to be grown in living tissues. The second thing is to induce disease from a pure culture, an animal has to be susceptible to that pathogen. Some animals are susceptible to certain pathogens when other animals are not. And then another exception is some diseases occur only when they get the opportunity, right? We have Staphylococcus aureus on our skin, and typically it doesn't harm us, right? It's part of our normal flora. However, if we get a cut or a scratch that breaches our skin, then Staphylococcus aureus might take that opportunity, right, to become pathogenic to us. And so these postulates, we still use these today, and the work of Koch has, if he hadn't done this work, we wouldn't be where we are with our advances in our understandings of microbiology today. Other things that he developed, the pure culture technique, also developed an auger that was a medium that he could grow bacteria in. He also developed the streak plate method, and he later on was actually given the Nobel Prize for Physiology or Medicine in 1905. And he also went on to do some work regarding tuberculosis and cholera and syphilis. And so his students also kind of carried on with Koch's work and continued work with tetanus and diphtheria and syphilis. And one of his students actually received the Nobel Prize in 1908 and was given the name of the father of chemotherapy. So now we find ourselves in the modern era. So this is microbiology as we know it today. And as the 20th century progressed, there were so many discoveries in microbiology, bacteriology, virology, and genetics. And within 100-year span of time, which is a relatively short time if you think about it, there were many discoveries and advances that have literally changed our entire lives. And so, we don't need to memorize these, but I did pick a few that I thought would be interesting. And the first one is in 1928, the discovery of the first antibiotic, right, by Alexander Fleming. He noticed a mold, and he later identified that as Penicillium notatum. And then in 1942, germ warfare. And the Japanese actually released insects coated with diseases like anthrax, cholera, typhus, and bubonic plague, and that affected approximately 300,000 people. And then in 1946, we discovered that bacteria could do this amazing thing called conjugation. And conjugation is the coupling together, or the process in which genetic information is transferred through cell-to-cell contact between bacteria. Bacteria are sneaky little creatures. We're going to learn that as we kind of dive into this a little bit more. And then in 2001, bioterrorism, thanks to our good friend anthrax. And then, just most importantly, not memorizing these dates, but just learning from our past and, where it's brought us to and how resourceful the microbial world is and its survival methods, because the organisms are so simple. It takes a lot for the human organism to be able to change and evolve. But for these little creatures, they are extremely resourceful at being able to adapt and overcome and evolve. As a surgical technologist, you are going to come in contact with every single type of body fluid that a human has. And in that regard, we need to use something called standard precautions. Now, you may have heard of universal precautions. Well, standard precautions is a combination of universal precautions and body substance isolation. Basically, standard precautions say that any type of body fluid, whether it's blood, semen, sweat, secretions from mucous membranes, urine, any of those fluids could possibly contain transmissible infectious agents. So the special section within the CDC is called the Healthcare Infection Control Practices Advisory Committee, or HICPAC. And the members of this committee, they're responsible for making and reviewing guidelines, advising the CDC and the U.S. Secretary of Health and Human Services and other agencies regarding a broad scope of public health issues. And, these standard precautions, they're going to apply to all patients, whether it's baby or grandma or anybody in between. And so some of these practices include washing our hands, using gloves, using gowns, using masks, using eye protection and face shields depending on the anticipated exposure, and safe injection practices. Also, equipment should be handled in the same manner, right, to prevent transmission of infectious agents. So whether that is wearing gloves so that you avoid directly contacting it, making sure that soiled equipment is clean and disinfected in a specific way, some new practices that have been added to the standard precautions are in the areas of respiratory hygiene and cough etiquette, safe injection practices, and the use of masks for when we are either inserting a catheter or injecting some sort of material into the spine or epidural space. And then there's transmission-based precautions, and there's 3 types of those. There's contact precaution, droplet precaution, and airborne precaution. And we use these when a specific disease process or something that is harmful and pathogenic to humans has been identified, and those other practices that we use does not address the mode by which it is spread. Okay, so some diseases we have to use more than one of these transmission-based precautions, like TB. TB is airborne precaution and droplet precaution. So sometimes we're using more than one, but overall we want to pretend like everybody has something that we don't want to get and take home to our family. So I'm going to get up on my soapbox for a minute and I'm going to talk about something I'm very passionate about, and that's surgical conscience. And our surgical conscience is defined as the honesty and moral integrity that the surgical technologist must possess in order to always practice strict aseptic and sterile techniques, to not hesitate to admit a mistake or break-in technique, and take corrective actions to prevent potential harm to the patient from acquiring a surgical wound infection or any other preventable injury. And that is a big responsibility that we have. And first and foremost is our practice of aseptic technique. And we're going to be going through these in this class. And if any of you out there have ever played a sport, and all sports have these specific rules to them, well, if surgery was a sport, our rules would be our sterile technique, right? Our principles and practices. So we're going to learn those. And first and foremost, that's the easiest way that we can protect our patient. And so surgical conscience is doing the right thing, even though nobody saw you do something wrong. Because at the end of the day, whatever that is going to impact your patient in a negative way. So we always want to make sure that we are practicing the best sterile technique, and if we make a mistake or a break-in technique, or we see somebody else make a break-in technique, that's a big one too, right? It's not just about holding ourselves accountable. Sometimes we need to hold others accountable too. So as the surg tech, you need to be brave enough when you see the surgeon's hat contaminate the sterile light handle, or if you see a little hole in his or her glove, to be able to say, hey, we need to change your glove. Stop, you have a hole, let's change that, right? And to be able to fix it. No matter how long you're in this field and you're scrubbing, you're still going to make breaks in technique. You're still going to contaminate things, and hopefully it's not as often as when you're starting out brand new, but you're still going to. And I tell everybody that these things are going to happen. It's okay. It doesn't define you. What defines you is what you do when you realize you have made a break-in technique or a mistake has occurred. That next best thing to do, that's your surgical conscience speaking up, saying, hey, we need to fix this and make it right. So that concludes our lecture for Chapter 1. I told you microbiology was going to be fun.